few years is an interesting description. Started with 2004 with this. Yeah.
This is tree time.
Yeah. Mm-hmm. Yeah. Unfortunately, you have to listen to me a couple of minutes more, but Jimmy will take over. So be patient. Yeah, we have been working with this greater Capricorn beetle. It resides in this nature reserve that we just saw, Halltorpshage, and it's the only site in the Nordic countries. It used to live in the Swedish mainland until the late 60s, and then it got extinct. So since the population in Halltorpshage is so small, and the number of trees, as we just saw, are also very few, we realized that we would need to reintroduce this species into areas where it used to live. And in that process, of course, it's very important that you have knowledge about the area where you reintroduce the species, because we have many sort of enthusiastic projects where you breed up a species and put it out and it dies out again. So of course you have to have thorough knowledge, and in Sweden we have that because we have made these huge tree inventories. For the greater Capricorn beetle we have chosen two areas in Kalmar and Blekinge County where we have a lot of these old oaks that are standing in good conditions, I should tell also. Because if we had very old oaks standing in very dense forest, it wouldn't have helped this species. Okay. So we have had this management plan, the action plan, national, published by SEPA regarding these species, and you see this was published in 2010, but we started the actual work 6 years earlier. And then Life Bridging the Gap came, so we could get funding for starting the reintroduction. And this is sort of an evening in Halltorpshage. So these two trees are, that you see that the old oaks, they are colonized by greater Capricorn beetle. And you could see the signs of the species in the bark, like very large holes that you could put your thumb inside. And we can also say that this species has been also debated. Some species says it's a pest species, and recent research shows that it's, on the other hand, could be a key species, that it creates habitats for other species, for example bats and insects, probably also fungi. And I should say also in Halltorpshage, since we have so much control of the trees, we could see that some trees have been colonized by Cerambyx cerdo, but the oak has had better condition with a lot of tannins and so on, has thrown Cerambyx out of the oak. So it's not like if you have Cerambyx in an oak, the fate of the tree is closed, so it could go either way. So we have made surveys of the number of entrance holes up to 4.5 meters. Seems to be sort of a figure, 4.5 meters. It's the end of the ladder. But it's a relative thing that we could see that this population has never reached 50 individuals. It's more or less staying in 20 to 30 individuals per year, so it's a small population. And then you could see the orange staples here. It's a number of old oaks that you have Cerambyx in, so it's normally 3 or 4. And then you have the gray staples that shows the number of trees that are in sort of bad condition that this species could use for a couple of years. And now, Jimmy, it's your turn.
Okay. Great, thank you. Thank you,
Life Bridging the Gap, for having me. My name is Jimmy, and I work at a non-profit conservation organization called Northern Ark. And we were the ones who were appointed Jonas' crazy idea to try and breed this species. And the project started in 2012, and our aim with it was to develop a successful breeding method on how to rear the species in captivity. So since the population was so fragile in Sweden, we wanted to start with a Polish population instead, because the species is much more healthier there. And we had permission to collect 15 pairs a year for 3 years. Here you can see Jonas in a dodgy hotel room looking at beetles that we have collected that day. And the beetles that we did collect were transported back to Sweden in cooling boxes so that they wouldn't burn too much energy in the journey. And at Northern Ark's breeding facility, the beetles were put in pairs. And each box was furnished with 2 pieces of oak that had thick bark for the females to lay eggs in. And the beetles were also offered a lot of different foods. They seem to like sweet food, so they really like banana. They eat pear. They eat nectar juice that we give them. And Jonas and Professor Claes Andrén, who usually used to work at Northern Ark, did this mixture that you see in the bottle there. That's apparently wine, beer, and banana. And I was told to feed them that. Yeah. And so I did, because I'm obedient. And that didn't turn out as well as we thought. I'm quite happy, actually, because the room more or less smelled like the back of a bar. But we were still successful, and we monitored all the behaviors very carefully. And when the first eggs were laid, we then collected these and put them into petri dishes with a bit of moist paper inside. And about roughly 2 weeks later, the first eggs start to hatch into tiny larvae. And a newly hatched larva weighs around 0.04 grams. This is obviously still a big species, but despite that, the larva is very small. All the larvae are then kept one by one, because this species, like many other saprophilic species, are opportunistic carnivores, so they will gladly eat each other if they have the opportunity. Over time, you won't find all the eggs, and they will start to hatch inside the wood. And then we have to collect the larvae that has started to chew away inside there. It looks quite invasive, but it's actually easier than it looks. So most of the larvae can be collected without damaging them. And I would say at the end of the year, you can find 30 to 50 larvae in each piece. We, of course, didn't know what to feed these. So we did a bunch of trials where we tested different foods. Or the challenges that we met were to, of course, create a food that the larvae liked and accepted. We also wanted to make a food that showed a nice growth rate. One important thing is also, when doing this in a large scale, to make it easy to work with, and if possible, not to make the food mold as quickly. Because when organic material meets moisture, you will have mold. I won't go into very much detail here, but these are just some of the tests that we did early on. And up in the corner there is the recipe that we then went with. We have now refined it. But it consists of sawdust and mixed with commercial Drosophila food. Mixed with some water. And as you can see, the larvae grew very well. The other tests here, the larvae either stopped growing or eventually perished. So now we could do that quite successfully. And this video is obviously speeded up a bit. I think it's 15 seconds when we see it, but in reality it's almost an hour long. So when I look at them, I never see any movement almost. But as seen here, they actually have a very good idea of what they do when they move around the food, and so it's interesting to see. When the larvae is approximately 3 centimeters or more, then they are big enough to change skin for the last time and turn into pupa. And this process goes very quick, takes about 20 minutes, 30 minutes or so, until the pupa is fully developed. You can even see the antennas getting longer and longer. And in this stage, it's possible for us to see if it's a male or a female already. So the male has the longer antennas, and the female's antennas meet down at the abdomen. One very important thing for this project was to find out how to care for the actual beetle. We did several trials where we hibernated newly hatched beetles and also actually even hibernating pupas to see what was the most successful way of doing it. We soon found out that a winter period for the newly hatched beetle is vital for them to be able to mate. So without hibernation, they won't be fertile at all. In our breeding population, the life cycle from an egg to a finished beetle takes 2 to 3 years, depending on how quick the first larvae grow. Sometimes they develop after 2 years, sometimes 3 years. Naturally, we say that it takes 4 to 5 years. I don't know how accurate that number is, but if it is, then we have shortened the period quite a lot. And as a side effect to this, the beetles end up a bit smaller. So instead of being 5 centimeters plus or so, they are, some of them are 3 centimeters. However, they are still very much indeed reproductive. But a larger beetle will of course lay more eggs. These still lay a lot of eggs, so we're not very worried about that. In 2015, 3 years after we started, we were able to rear the first F1 generation. So that's when we felt secure enough to start with the Swedish population. So the now quite big Polish population that we had in captivity, I think it was even 1,200 individuals, were then delivered to Poland again, and actually to Wrocław Zoo, who then reared them and released them in and around the zoo. I don't have any information on how that went, but they did anyway. So then we have to start all over again, more or less, with creating a new ex situ population. And we did so using breeding material from Halltorps hage. And here we had permission to collect 5 pairs a year for 3 years. And the beetles were only on a loan. So they were brought to Nordens Ark, and the females were allowed to lay as many eggs as they could for a month, and then they were returned so that they can continue to lay eggs in the reserve. We never found the full amount of beetles, as Jonas' slides showed. The species is quite rare in the area, despite the area being quite small. Nevertheless, with the beetles that we found, we have now been able to build a very nice captive population still. So, after the first 5 years, we finally had the first beetles ready to be released in Sweden. And with the method that we now have, we can produce between 200 and up to 350 beetles roughly. And this project has been very interesting to other conservation initiatives as well. So we have had visits from Germany, Austria, Denmark, who has learned how we do this in order for them to work with the same species or even with other endangered species. And I think that's something to be proud of. Like Jonas said, the introductions have been focused to 2 nature reserves, one in Kalmar County in Björnö, and another one in the very south of Sweden in Tromte. And this is how, this is how the cages look, or how it can look. So some of the trees are fitted with these netted cages, and 2 to 3 pairs are let in to each cage. This is to protect the beetles from predation, but the main release is just done with beetles placed on the trunks, and those beetles are then able to fly away and find whatever oak that they want to lay their eggs on. We've also been able to use this captive population for other interesting studies, and pheromone studies is one of those. We have done this together with the Swedish Agricultural University in Alnarp. It's not finished yet, but as you can see on the chromatograph staples there, we think that we now have what is supposed to be the attraction pheromone for the male. That staple actually goes way above the slide. And if we're correct, then this will be produced artificially and then in the future used to survey for the species. We've also worked quite a lot with training a detection dog to find larvae of greater Capricorn beetle. And the dog in the picture is called Luca, and her owner and trainer Elin, who works at Nordens Ark, can now easily detect greater Capricorn beetle from other longhorn beetle species. It's quite astonishing, actually. Hopes are that we can use the dog already this year. This will hopefully also make it easier for me and Jonas and whoever who wants to look for the species to find out where they go and which trees they have inhabited. This is not my picture. I borrowed this from Hollow Hill. I think it's from a radio telemetry study made in the Czech Republic. And this summer, in 2 weeks actually, we will do the same thing where captive bred greater Capricorn beetles will be fitted with these. 50 of these actually. And hopefully this will tell us how long the beetles live. Where do they go? How long do they disperse? Because as for now, there are a lot of unsolved questions with the released beetles. This year could potentially also be the first year when we find the first offspring in the wild. So we're very excited about that. I think this is my last slide. I didn't go over time even.
No.
Wow.
Definitely.
If you have any more questions, then I'm up for that.
Okay, thank you very much.
Thank you.